It's hard to imagine our daily life without milk. It is an important nutrient and forms the base of all our dairy products. Daily, countless litres of milk are produced and consumed worldwide. However, raw milk does not simply enter the dairy chain just like that. The milk has to meet strict standards to make it suitable for the production of first-rate dairy products and to rule out all the health risks to humans and animals. In order to ensure excellent quality products, the Dutch dairy chain is continuously monitored by Qlip. Qlip is an independent organization serving both dairy farmers and the dairy industry. It has established a unique position in the dairy chain. Worldwide, Qlip is among the largest processor of dairy samples for dairy herd improvement organizations. Qlip is a trendsetter, both with its facilities for raw milk testing and for the analysis of dairy product samples. The same applies to systems for quality assurance in the dairy chain that have been developed through the years in close cooperation and alignment with the sector. The core business of Qlip is the analysis of raw milk and dairy products on assignment to dairy companies and dairy herd improvement organizations. The auditing and certification of the processes at the farm, during transport, and at the dairy plants. In addition to cow's milk, Qlip also tests goats and sheep milk. All services are carried out efficiently and in conformity with international standards. The internal quality system is officially ISO accredited. Qlip cooperates closely with the dairy industry and is continually investing in improvement and innovation so it can guarantee meeting the needs in the chain now and in the future. Qlip focuses on speed to ensure that the results are received by the customers as quickly as possible. They also ensure reliability, cost leadership, and dedicated services by applying innovative information technology. The dairy farmer delivers milk to the dairy company. Payment by the dairy company to the dairy farmer is based, amongst other things, on the tests for composition and quality carried out by Qlip. The milk is collected from the dairy farm every 3 days by a milk collection truck. The driver takes a sample from the farm tank using a sterile plastic bottle, which is fitted with a data chip. A GPS system determines exactly at which dairy farm the tanker is situated, and the chip is accordingly loaded with the relevant data. Amongst others, this data consists of the farm tank number, date and time of collection, the receiver of the milk, and the ID of the milk collection truck. All samples are collected and stored at the dairy plant, from where Qlip transports the cooled samples to its laboratory. Qlip operates an own transport service. The samples are collected throughout the Netherlands twice a day, 6 days a week. The whole system of sample storage and transport is part of a highly controlled cooling chain, which safeguards the quality of the samples. This is extremely important in obtaining correct test results. Thousands of samples arrive at the payment testing laboratory every day. A robotic arm places the bottles with their chips into special carriers, which are then transported along a conveyor belt. The Qlip computer system registers the samples and loads the plan of analysis for each individual bottle into the chip. Then the journey through the laboratory can start. The test results are the basis for the payment to the dairy farmer by the dairy company. The composition of the milk is tested according to fat, protein, and lactose content, with the aid of infrared equipment. In relation to quality, several parameters are frequently determined. The bacterial count is a value for the number of bacteria in the milk. The somatic cell count provides information on the udder health status of the herd. The determination of free fatty acids serves to monitor the functioning of the milking equipment. The freezing point mimics eventual adulteration with water. The samples are also tested for the absence of antimicrobials and of butyric acid bacterial spores. The latter is indicative for the hygienic quality of the silage that is fed to the cows. Note, the farmers receive additional information for management purposes. Urea data indicates nitrogen utilization on the farm, and testing on chloroform elaborates the practiced care in cleaning of the milking equipment. In the context of animal health control, Qlip analyzes herd bulk milk samples on antibodies against Salmonella. Qlip reports the results of most of these tests to the dairy company within a day after sampling at the farm, so that the dairy farmer has the results of the analysis at hand. After use, the bottles are stripped of their chips and shredded for recycling purposes. The chips are reformatted and then ready for reuse. By participating in milk recording, a dairy farmer has the choice of a variety of options. He can choose from a number of different sampling frequencies, whether he takes samples himself or has them taken, and which types of tests he wishes to be carried out. At the farm, the milk samples are stored in a lockable sample box. The farmers receive sufficient bottles with preservative. These bottles are placed in a vial carrier, which is fitted with a data chip for identification. After sampling, an employee of the Dairy Herd Improvement Organization collects the filled bottles in transport boxes and brings these to one of the regional collection centers. Qlip takes care of the transport from the collection centers to its laboratory in Zutphen. Qlip analyzes samples in accordance with international guidelines. In order to further optimize the benefit of his operations, a dairy farmer can make use of dairy herd improvement services. Frequent analysis of milk samples from individual animals provides useful data on productivity and udder health within his herd. At the same time, this data is effectively used for dairy cattle improvement in a wider sense. After registration, Qlip analyzes the milk samples for fat, protein, lactose content, and urea values with infrared equipment. Furthermore, the individual somatic cell count is determined, by which invisible problems in udder health can be spotted. At the same time, farmers receive urea data that indicates the nitrogen utilization of the cow. Quality is key for Qlip when analyzing samples of milk. Reference samples with known values are frequently added to the conveyor belt. And part of the samples are re-examined to see whether the differences in results are within limits. After regular analysis, Qlip offers the option to have milk recording samples selected for more specific examination, amongst other things, to monitor animal health. These samples are collected at a separate conveyor belt. Later on, the milk will be pipetted into special blocks. After use, the bottles are emptied and shredded for recycling purposes. The chips are reformatted and are then ready for reuse. After cleaning of the vial carriers, new bottles supplied with preservative are put into them. The refilled vial carriers are put in the transport boxes and are then ready for transport to the regional collection centres of the dairy herd improvement organisations.